My name is Till Luckenbach. I'm from the UZ in Leipzig in Germany. Welcome to my talk, Transcriptomic Effects of Chemical and Temperature Stress in Amphipods from Lake Baikal in Siberia. The data I'm going to present are from the project Lake Baikal and the Effects of Global Change with the partners UZ, Alfred Wegener Institute for Polar and Marine Research in Bremerhaven, Irkutsk State University in Russia and the University of Leipzig. Lake Baikal is so big you can see it from space. It is also called the Sacred Sea, although it is a freshwater lake. It is so unique that it is a UNESCO World Heritage Site. Baikal is a global biodiversity hotspot. More than 2,600 species have been identified. Most of them are endemics, and the amphipods are a prominent taxon of Baikal with 350 described species. All of them are endemics, and they make up about 45% of all known freshwater amphipod species. The water conditions of Baikal are very unique. The water is saturated with oxygen along the entire water column, and Baikal is very deep, so it goes down to 1,600 meters. Water is continuously intermixed, and the water is cold with 6 degrees mean temperature, and it's super oligotrophic. Its fauna is completely distinct from other freshwater environments in the northern hemisphere, so even ponds in its direct vicinity are inhabited by a fauna that is different from the Baikal fauna, and it's more a typical Palearctic aquatic fauna. We addressed the question whether this species immiscibility barrier between Baikal and other aquatic environments may get breached by global change. So we pretty much started off with the assumptions that Baikal amphipods are highly adapted to specific, pristine, stable conditions, whereas amphipods that are ubiquitous in the Holarctic are adapted to conditions within a wider range and more adapted to fluctuations of conditions. So Baikal amphipods, we assume, have no or little cellular stress response capacity, whereas other amphipods have a more pronounced cellular stress response capacity. So if conditions now change with global change, then these other amphipods that don't inhabit Baikal yet may be able to invade the lake and outcompete Baikal amphipods. The species that we study are Baikal species Eulimnogammarus verrucosus and Eulimnogammarus cyaneus, and Gammarus lacustris, which is pretty much ubiquitous in the northern hemisphere. It also occurs in the Baikal area, but not in Baikal itself. For the experiments, we sampled the animals in the field, brought them to the lab, and exposed them to cadmium chloride as a chemical stressor and temperature stress for up to 24 hours. Then the animals were sampled, frozen RNA was extracted, and sequenced on a next-generation sequencing device. The genomes of those examined species are not available, so de novo transcriptome assembly had to be done, and these assemblies serve to map the reads to identify genes that were differentially expressed. Sequence annotation happened by comparing the sequences that we got with the sequences in the NCBI database. We used gene ontology terms to look for gene categories with differential gene expression. So this is an overview of the annotated transcript sequences that we found. So depending on how we look at it, 10 to 40% of sequences were from metazoa, so amphipod sequences. There were a lot of ciliophora sequences, which is because those are common commensals of amphipods. And then also sequences from other taxa, that were probably also commensals or parasites. And a lot of sequences were not, could not be annotated. Now, upon exposure to temperature stress, you see that we found genes that were down and upregulated. So this is an overview of the number of genes that were differentially expressed. Most differentially expressed genes were in Eulimnogammarus verrucosus, less in Eulimnogammarus cyaneus, and the least in Gammarus lacustris. That was similar upon exposure to cadmium chloride. So most expressed genes, differentially expressed genes in Eulimnogammarus verrucosus, less in Eulimnogammarus cyaneus, and least in Gammarus lacustris. Generally, the numbers of differentially expressed genes were higher after 24-hour exposures. So this shows GO terms, and in the upper panel you have the upregulated genes, and upregulated genes upon temperature stress belong mostly to cellular stress response GO terms. So in the lower panel you see downregulated genes which were from GO terms related to cellular function, like translation, transcription, and so on. And we got a similar picture with amphipods exposed to cadmium chloride. So again, upregulated genes belong to GO terms related to temperature stress, stress response, and downregulated genes were from GO terms related to cellular functions. So these data indicate that upon stressor exposure, the energy is redirected from cellular functions to cellular stress response. And what was interesting was that the bical amphipods do have cellular stress response capacities, so pretty strong. That was pretty high. Whereas it was surprising that Gammarus lacustris showed such a little response to the stressors. And we assume that this may be related to the experimental temperature, which actually occurs in the environments of Gammarus lacustris. So it does survive at this temperature, but it may have led to a depression of the transcriptomic response to stresses. Now, another finding in the transcriptome data helped us to explain earlier findings that we had, which was that the HSP70 response in these amphipods was always quite weak, which was really surprising. So here you see qPCA data related to RCX levels of cadmium chloride, which was used as an inducer of HSP70. Cadmium concentrations we equated with RCX levels, which is representative of the stress level caused by the cadmium. And you can see that up to LC50, which is already pretty high stress level, there were maybe statistically significant, but overall little changes of transcript levels. Yeah? This was always surprising because HSP70 is supposed to be very sensitive, and it is supposed to respond strongly to stress. Now, analysis of the transcriptomes of the stressor-treated amphipods showed that there were various more or less induced HSP70 forms in the amphipods. And this is a phylogenetic tree, and you can see there are lots of HSP70 forms there. Some of them are inducible, some of them are not inducible or less inducible. So it's also a question which forms we actually caught with qPCR. The inducible and non-inducible HSP70 form could be distinguished by certain amino acid signatures. Now, finally, in this transcriptome study, we also had a treatment with phenanthrene, which I didn't mention before, because the results were kind of puzzling that we got. So we use as a solvent acetone, and because we thought we had a very low concentration of the solvent, and then we thought it gets it evaporates immediately, but indeed it showed a very strong effect on the transcriptome. So yeah, this is an interesting finding and should be considered in future experiments where transcriptome studies will be done. So don't use acetone as a solvent. Большое спасибо. Thank you very much for your attention.